Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Hope so you all are good, fine and healthy. Well, today in the previous video we have discussed about the prophase of mitosis. Now we are going to discuss about the metaphase and the anaphase. So let's see what is happening in these steps. Students, as you know that the mitosis have four major phases: prophase, then metaphase, then anaphase, and then it's come telophase. Well, student, what is happening in the prophase, which we have already discussed, the thread-like structure going to conduct itself in the form of the chromosomes having the sister chromatin and the centromere. The nucleolus and the nucleus used to degenerate, nuclear membrane used to degenerate, while the centrioles present in the cell after duplication move towards the opposite pole of each other. And they will start producing the spindle fiber, and these spindle fibers altogether is known as the mitotic fiber. Then the chromosomes are going to, their kinetochore protein is going to attach with the spindle fiber, and they are going to arrange themselves in the center of the cell. All the chromosomes are going to arrange themselves in the center of the cell, and now this is known as a metaphase plate. So let's see what is happening in the metaphase. Well, students, before going in detail, you should know about the kinetochore fiber. What is basically the kinetochore fiber? The kinetochore fibers are those spindle fibers which are going to attach with the kinetochore protein of the chromosome. The fiber, the spindle fiber, which is going to attach with the kinetochore protein, it is known as the kinetochore fiber. The spindle fiber, students, listen to me very carefully, the spindle fiber which is going to attach with the kinetochore protein is known as the kinetochore fiber. While there is also, students, there is also a non-kinetochore protein fiber. The non-kinetochore fiber is that fiber which is not going to attach with the kinetochore protein of the chromosome. The spindle fibers which are going like this, let's suppose this is a centriole. These are the centrioles which are at the opposite side of the cell, which are the opposite side of each other. The fiber which is going to attach with each other instead of the chromosome is known as the non-kinetochore fiber. Those fibers which are going to attach with each other is known as the non-kinetochore fiber instead of the chromosome. A fiber which is going to attach with the kinetochore protein of the chromosome is known as the kinetochore fiber. But a fiber which is the spindle fibers which are going to attach with each other instead of the chromosome will be known as a non-kinetochore fiber. So, two kinetochore fibers from opposite pole will attach with each chromosome. The two kinetochore fiber will attach with each chromosome from opposite pole, like one from this pole and one from this pole. One from this pole and one from this pole. This means that each chromosome kinetochore protein is going to attach with the fiber from opposite sides, from both opposite poles, one from this pole and other from this pole. Now, after the joining of the spindle fiber with the kinetochore protein, the chromosomes will start arranging themselves in the center of the cell. Now, what is happening, student? All the chromosomes will arrange themselves in the center of the cell. After joining with the spindle fiber with the kinetochore protein, the chromosomes will arrange themselves in the center of the cell, and this will be known as a metaphase plate. There will also be the non-kinetochore fibers which are also going to attach with each other instead of the chromosome. So, the chromosomes are going to arrange themselves in the center, and this will be known as a metaphase plate. These steps are going to happen in the metaphase. In the metaphase, the spindle fibers are going to attach with the kinetochore protein of the chromosomes, and then chromosomes are going to arrange themselves in the center of the cell, and this is known as a metaphase plate. The next phase will be the anaphase. Students, here is the image of the metaphase. This is basically the metaphase plate. The chromosomes have arranged themselves in the center of the cell, while the fibers which are actually joining with the kinetochore protein are known as the kinetochore fiber, while those fibers which are attaching with each other instead of the kinetochore protein are known as a non-kinetochore fiber. So the nuclear envelope and the nucleus is degraded in the prophase. Now let's see what is going to happen in the metaphase. The centrosomes, as I told you before, they will start to produce the spindle fibers, and now the chromosomes, like these spindle fibers, are basically going to attach with the kinetochore protein of a chromosome. Well, this is a chromosome and the red is basically the kinetochore protein. Now here, student, you can see clearly that there are some fibers which are joining with each other, and these fibers are basically the non-kinetochore fibers, and while those fibers which are attaching with the kinetochore proteins are known as the kinetochore fibers. After these spindle fibers joining with the kinetochore protein, the chromosomes will arrange themselves in the center, and this center will be known as a metaphase plate. In this video, you can see it clearly that the spindle fibers are actually producing by the centrioles are going to join the kinetochore protein, as well as there are also some fibers which are joining each other. The spindle fibers which are joining each other instead of the chromosomes are known as a non-kinetochore fiber, while after attachment of the spindle fiber with the kinetochore protein, they are going to arrange themselves in the center, and this will be known as the metaphase plate. Well, what is now happening in the anaphase? Listen and see the video very carefully, and observe what is happening. So students, after the metaphase, the anaphase begins. Here you can see what is going to happen. The spindle fiber from the centrosomes from each pole are now going to pull the chromosomes toward each other. And now what is happening? Each sister chromatin at each pole. Each sister chromatin in each pole. It means now there is a suppletions of the chromosomes. You can see that now each pole will have equal number of the chromosomes, as well as the non-kinetochore fibers are also elongated themselves. In anaphase, students, these sister chromatins are going to get separate after the pulling of the kinetochore fiber toward its originating centrosome. These fibers have originated from the centrosomes, and these are going to pull the chromosomes toward themselves. Now you can see now there are two sets of the chromosomes, and sister chromatins are now divided. One set to one side and other set to other side. And you can also see that the non-spindle fibers are going to elongate themselves. After that, the telophase begins. But first, let's discuss in detail about the anaphase. Well, student, this is basically the anaphase. You can see here that the centrosome, basically the centrosome is now going to pull the kinetochore protein toward itself. The centrosome, basically, the kinetochore fiber is going to pull the kinetochore protein towards its originating centrosome. So what is happening here, student? Basically, the kinetochore fiber is going to pull the kinetochore protein towards its originating centrioles. Basically, now as you know that together they were the 92 chromatin. After the division, what will happen? Each set will have the 46 chromosome. Here are 46 chromosomes, as well as here are now 46 chromosomes. And the chromosomes will be like in this form. A single chromatin, a sister chromosome only, a single chromosome to each set. Now, after the anaphase, the next phase is the telophase. Now, what is going to happen in the telophase? Basically, student, if we are going to discuss about the telophase, then telophase is totally reverse of the prophase. In the prophase, what used to happen? In the prophase, the spindle fibers start producing, so these spindle fibers are going to disappear. Similarly, the nuclear membrane will form as well as the nucleolus will also form. The nuclear membrane and nucleolus will form. Similarly, this chromosome will again convert themselves into the thread-like structure. So, you can simply say that the telophase is totally reverse of the prophase. In the telophase, there will be again conversion of the condensed chromosome into the thread-like DNA. The nuclear envelope will reappear as well as nucleolus present in the nucleus will also reappear. Moreover, there will be, you can see the spindle fibers will disappear themselves, and the cytokinesis will actually split these cells into these daughter cells into two cells. But right now, understand what is a telophase. After the anaphase, the next phase is obviously the telophase. So, in telophase, as I told you before, that is the reverse of the prophase. Basically, what is going to happen? There will be the reappear of the, you can say, nuclear membrane around this chromosome, and this chromosome is going to again convert into thread-like structure, and nucleolus will appear again as well as nuclear membrane, and centrioles will stay there. After that, the cytokinesis begins. Basically, beta cytokinesis, as I told you before, is the division of the cytoplasm. This cytoplasm will divide, and now there are two daughter cells. The cytokinesis occur by the process known as the cleavage. Now, what basically is the cleavage? The cells are going to separate by the process known as a cleavage, and for the point where they are going to cleavage themselves is known as a cleavage. Cleavage furor. In the cleavage furor, what is going to happen? Slowly and gradually, the chromosomes will actually get deeper, and the point comes that they both are going to get separate from each other. This cleavage furor will occur at the place where the metaphase plate were during the metaphase. The cleavage furor will occur at the point where the metaphase once began, where the metaphase plate were formed during the metaphase. This will be the point where the cleavage furor begins. Basically, beta, what is a cleavage furor? The furor, in the cleavage furor, what is happening? Slowly and gradually, the cytoplasm will get deeper, and the time will come that they both will get separate from each other. This is known as a cleavage furor. But in the case of the plant cell, it is different, because you know it very well. In the plant cell, there is not only the cell membrane, but also there is a cell wall. As well as more thing is that the plant cells do not contain actin protein, which is going to help in the cleavage furor. Basically, students, the animal cells contain the actin protein. This actin protein will help in the cleavage, but the protein is not present in the plant cells. So how they are going to do so? Let's see. Students, you can see here clearly the cleavage furor used to made in the animal cell by the help of the contractile ring or the microfilaments like actin proteins, which are going to help to form the cleavage, and this cleavage is going to separate the cell into two cells. Now, what is actually going to happen in the plant cell? Let's see. Students, basically in the plant cells, listen to me very carefully, what is going to happen. Let's suppose there are two daughter cells. Now, how these daughter cells are going to separate? Basically, the Golgi apparatus present in the cell will start producing the vesicles. The Golgi apparatus will start producing the vesicles, and these vesicles are going to arrange themselves in the center of the cell. These vesicles are going to arrange themselves in the center of the cell where the metaphase plate used to form. Now, more vessels are going to produce, and slowly and gradually these will form a complete line with the vesicles. The vesicles are going to cover this whole scenario. And then these vesicles are going to join with the cell membrane of the plant, plasma membrane of the plant, as well as now the cell wall will also start forming on that. Basically, this is known as a phragmoplast. This is known as a phragmoplast. So this plate will be known as a phragmoplast, and now after that the cell is going to split into two daughter cells. So student, listen to me very carefully. Basically, this is a plant cell, and the plant cells, obviously two daughter cells are there. Now the Golgi apparatus present in each cell will start producing. We are students learning about the cytokinesis, that how the cytokinesis will occur. Basically, the karyokinesis have completed. Now we are going to move, the plant cell is going to move toward the cytokinesis. In the cytokinesis, the Golgi apparatus will start producing the vesicles. The vesicles present in the cell will start arranging themselves in the center of the cell, named as the cell plate. Slowly and gradually, the number of the vesicle cells will increase that cell plate. And the time will come that cell plate will join with the plasma membrane of the plant cell. And now the cell wall will start forming on that side, and now it will separate the two cells. It will separate the two cells from each other. And now there are two cells. Two plant cells will be there after the formation of the phragmoplast. Students, basically the cytokinesis in the animal cells occur by the help of the actin and the microfilament, as I told you before. The microfilament and the actin protein used to form the contractile rings at the equator of the cell, where once the metaphase plate formed. These microfilaments are going to get, like these contractile rings are made up of actin and microfilament. These contractile rings, when they are going to get smaller and smaller, the cleavage will become deeper and deeper. The contractile rings will become smaller and smaller, and the cleavage will become deeper and deeper and deeper, and the point will come when these both cells are going to get separate from each other. But in the plant cell, it is not like the same. It is quite different. The plant cells, as you know, that they do not contain the actin protein. So there will be no contractile rings. There will be no contractile rings, as well as no microfilament or actin. When there is no actin protein, so how the plants are going to get different? Basically, the Golgi apparatus is going to produce the vesicles, and these vesicles are going to arrange themselves in the equator of the cell, and that will be known as a cell plate or phragmoplast. Now, slowly and gradually, this cell plate will extend its size and will join with the original cell wall or the cell membrane of the plant cell. After that, they are going to get separate from each other. So, this is the way the cytokinesis used to occur in the plant cell. Students, keep in your mind the prophase and the cytokinesis of the mitosis is different in the plant and animal cell. In the prophase, if you remember, the plant cell do not contain the centrioles. Similarly, in that case, and in cytokinesis, it do not contain the active protein. So that's why their cytokinesis and the prophase is different from each other. Jazakallah khair. Hope so you understand the things very clearly and you get each and everything. If you are not able to understand anything, then do text me, do comment me, or watch the video again, make the notes and send me the audio. Jazakallah khair. Assalamu alaikum wa rahmatullahi wa barakatuhu.